the scaffold, the way you actually do it, there is a basic framework and you randomize certain positions. Now, what you do is you build these very large libraries. So the libraries actually contain over 1 trillion members, and you then do a display technology which allows you to very quickly isolate a few thousand with both the biophysical properties you want and the affinities you want. One of the advantages, actually, for them to get on the technical side of working with scaffolds is that you can get to a new molecular entity within 12 months and probably get to an IND within maybe 24 months, which is usually a lot faster than you can do so with a monoclonal. And it basically comes down to the fact that with these very, very large pools of molecules, somewhere in there is a molecule with the right affinity and the right biophysical properties. It's just a matter of finding.
You usually have things like steric genes or other
We do, yes. So, sorry, so we're talking about difficult amino acids. Yes, so some of the molecules we have had methionines in the binding, and we take those out. And we also, they're expressed in E. coli, so you have to think about the N-terminal amino acid to make sure you get a homogeneous product. And we also do a lot of T-cell activation assays and do a lot of animal work to make sure that we've got rid of any predicted epitopes in those molecules. Of course, you won't know about immunogenicity until you go into humans, but you do the best you can with these T-cell activation assays. So we do some work. I mean, to be honest, out of the screen does not come the perfect molecule, but it takes not that much time to get a good molecule because you have such a small number of amino acids that you have to work with.
So my question is for Nick. You talked about improvements that need to be made in your area. And one of the areas was the route of administration. And I'm curious what role devices is going to play as you evaluate different technologies or as you try to minimize the impact of the route of administration concerns with that.
That's a great question. And we've actually discussed this as we were forming our panel. It's going to be product specific and indication specific. And so, for instance, we have some antibodies that we're developing for infectious disease. Ideally, we'd like those to be administered sub-Q. But in order for us to go forward, the development plan, at least within Morphotek, and everyone has their own plan, is at least to show product is active first. And for us, the straightforward way is to do IV administration. Once we get experience in man, we'll know if it's immunogenic. We'll understand what its PK/PD profile looks like, and then we'll be able to start our efficacy trials. Once we're in phase 2 and we have our hints of efficacy, then we start talking about reformulations. What would be the most beneficial to our patients? What would make it most compliant? Our lead program is a phase 3 trial in women who have relapsed ovarian cancer. It's a 900-patient global trial ongoing. The protocol is weekly administration by IV. Now, we're going forward with this to the marketplace. But we as a backup strategy, are now trying to figure out ways that we'll make this more amenable to patients to be able to have weekly administration versus going to the hospital for a 30 to 60-minute infusion, being able to administer that at home. I think the press release with Roche and Halozyme touched upon not only our philosophy but even Roche's philosophy, where you have maintenance therapy, chronic maintenance therapy, coming up with formulations that allow patients to have more beneficial exposure and treatment. In the case of some of the other indications, we have an antibody we're developing for broad anti-inflammatory responses. And here, one application is for asthma. It makes a lot of sense if we can do inhalation. However, many companies have tried to do inhalation with large macromolecules, which have made it much more difficult for its development. Having said that, the industry's grown since then. A lot more information has come forward. So we're looking at those types of opportunities. And then the last one is actually to downsize the molecule. And along with what Rob was saying, with regards to his scaffolds, we have the variable domains which are target specific. We could always cut the tails off of the antibodies to make them smaller and allow them to get into tighter spaces. So we continue to look at these other opportunities, but the normal development path is to get proof of concept with the canonical antibody first, then model what the market needs would be second.